Microbes are the most abundant organisms on the Earth's surface, and many of those microbes are going to be beneficial to us. So we definitely don't want to harm or decrease their overall growth. But in many cases, it is necessary to control microbial growth in the environment, in our food supply, on our body's surface, etc. So here we want to talk about some of the ways that we can control microbial growth in the environment specifically. Now, some terminology that does apply to controlling microbe growth is, first of all, sterilization. That word, if we're using it in its strictest sense, that refers to the removal or destruction of all microbes, every single one in a particular location. Now, that's not typically what we actually do. Instead, commercial sterilization is going to be the removal of harmful microbes. So if we're talking about harmful microbes, those are removed, and then there may very well be other microbes that are left, and they are still going to be surviving and maybe many times actually growing. Aseptic refers specifically to an environment that's free of pathogens. So that's definitely not talking about all microbes being removed from that particular environment. A lot of the beverages that we drink, you can see some examples in this image here on the slide. They are free from pathogens, but that doesn't mean that they're truly sterile. They're not free from every single microbe that would be inside there. Oftentimes, we're also culturing microorganisms, and when we culture those microorganisms on a petri dish, we are trying to grow specific ones and not grow others. So that would be a type of aseptic culture. If we talk about disinfection, disinfection is definitely something that happens in different workplaces, in the hospital perhaps, in our homes. And this refers to the use of physical or chemical agents, and the point is to inhibit or destroy microorganisms. So when we do this, if you look at just a couple of examples here, scrubbing the floor, the actual scrubbing part would be a physical agent. So scrubbing, we can also use different chemicals, maybe some bleach, maybe alcohol. These are not going to guarantee the removal of all pathogens, but this is going to be removing many of them, and so it's helping, and it's only used to refer to the treatment of non-living objects. So when we disinfect something, we're disinfecting a floor or a countertop or some type of eating utensil. Now, we also many times want to try to get rid or inhibit the growth of microorganisms on skin or living tissues. And when we're talking about inhibiting the growth on those living tissues, that's antiseptics. So antiseptics would be something that's used for that. And you can see the scrubbing and also the use of different detergents on the different medical personnel's arms when they're getting ready to do some type of medical procedure. Degerming is also another important word. This refers to removal by scrubbing. So we do have some of that does take place with the disinfecting that we just saw on the previous slide. Sanitation, another term, refers to the process of disinfecting places and utensils that are typically used by the public. So this is something that you do see in a lot of restaurants. So in their kitchen areas, they would be sanitizing dishes and different silverware, things like that. Again, a lot of different terms that we use to refer to this process of trying to control microbe growth to some extent. Most of the times it's not really true sterilization, because you will certainly still have some microbes on that particular surface, but we're trying to eliminate and get rid of the pathogenic ones and the ones that might cause us harm in the end. Pasteurization is a process that uses heat to kill pathogens and ultimately reduce the number of spoilage microorganisms in food and beverages. So we want to get rid of pathogens, but besides the ones that might cause us sicknesses and illnesses, we also want to get rid of the ones that would actually cause the spoilage of that particular product. If we look at a few suffixes that we do find on the ends of some words that pop up when we're talking about microbial control, the first one here is stasis or static. These mean pretty much the same thing, and these refer to a chemical or physical agent that's able to inhibit or decrease, really, microbe metabolism and growth. They're decreasing it, but they're not killing the microbe itself. The suffix "side" or "sidal" is referring to one that's able to actually destroy, kill, permanently inactivate some type of microbe. An example of that would be to say something is bactericidal. What that would mean is that it's able to kill or permanently inactivate bacteria. Microbial death is an important concept to understand when we're talking about controlling microbe growth. That term refers to the permanent loss of reproductive capacity under ideal environmental conditions. And if we plot the actual microbial death, as you can see right here, it is a constant. So we have this line that shows us a decrease in the microbial reproductive rate. And so we're looking at basically the number of living microbes on the y-axis, and that is a logarithmic scale. And on the bottom, we're looking at time. And we get basically a constant percentage of the population that is being killed at each moment in time, or each minute in time. Now, antimicrobial agents, so things that are against microbes, are going to function in two main ways. The ways that they do this is that they either disrupt the integrity, the composition, the intactness of the cell walls and the membrane of the cell, or the second thing that they can do is they can disrupt in some way the overall cellular metabolism and reproductive processes. And if we have either of those, that is going to disrupt the overall growth and reproduction of that particular organism, and in many cases, it will lead to the overall death of that organism. So if we talk more about the first one, the disruption of the integrity, the intactness of the cell walls and the membranes, well, first of all, we have to understand what those actually do. The cell wall is just this rigid box that's around the outside of the cytoplasmic membrane, and its overall job is to function in counteracting the effects of osmosis. What that means is that if water were to rush into the cell, then that water rushing in would typically cause the cell to swell up. And without the presence of a cell wall, that could cause that entire cell to just rupture, or we would call that lysis. So that would be deadly for a cell, and the cell wall is going to prevent that. But if we disrupt the integrity of the cell wall, then it may actually happen that too much fluid, too much water actually goes in, and you could get a ruptured cell. The membranes, they are very important for controlling what gets to go in and what gets to go out of the cell. And so if we disrupt that, you might get extra stuff flowing back and forth across that membrane. Some viruses, they contain envelopes on the outside, and disruption of those particular envelopes is going to inhibit their overall ability to attach to the host cell. And if they cannot attach to the host cell, then that would disrupt their overall life cycle. The second one we said was disruption of cellular metabolism and reproduction. This is most often accomplished by denaturing of proteins and destruction of nucleic acids. When we talk about denaturing of proteins, just as a quick review, denaturing is really referring to the process of unfolding them, disrupting their overall three-dimensional shape. And when we do denature them or unfold them, we are basically inactivating them. So we are taking proteins which have a whole long list of functions inside cells, enzymes, transport proteins, communication molecules, taking those and inactivating them. And nucleic acids, they contain the genetic information to make a variety of molecules inside the cell, and if we do destroy those, then that will inhibit the cell's ability to actually make the products that it does ultimately need to function. So when we go through and we actually try to select which microbial control methods are going to be used for a particular scenario, there are important factors that we do have to consider. One of those is going to be basically what is the nature of the sites that we do want to treat and get rid of microbes. The second one is going to be the degree of susceptibility of the microbes involved. Are they really resistant? Are they ones that are easily killed? And then the third one is going to be that we have to take into account the specific environmental conditions that we are dealing with. So when we look at all three of these, we're going to combine them, take them into account, and then use the best control method that we can come up with based on our specific scenario. So if we talk about the nature of the sites that are going to be treated and how that factors into all of this, first of all, we need to know how sensitive or fragile is the specific location. Because if it is something that's very sensitive, if it is very fragile, then we don't want to use a chemical or an agent that's going to be very, very harsh and destroy that particular site or location. Another thing that we need to know is, will the outer defenses be permanently damaged by accessing the site? This is when a lot of times if we want to treat something that is underneath the surface of our skin, to actually get to that particular location, we might have to make an incision. We might have to puncture the surface. And when you do something like that, you are creating a new wound, which could potentially be susceptible to infection. So we do have to take that into account. If we talk about the relative susceptibility of the microorganisms, some microbes are certainly more resistant than others. And if you look at the diagram right here, it does show you in general how they kind of line up with each other. The most resistant things are going to be prions and then your endospores. And then you can see that we have a whole list down to the most susceptible, which are going to be things like your gram-positive bacteria or enveloped viruses. So if we have something that is really resistant, we're going to have to use some tougher techniques. So bacterial endospores, as an example, these are capable of surviving very, very harsh conditions, and that might be very high temperatures, that might be high pressure, so it might take a combination of methods to treat them. If we're talking about the mycobacteria, these are unique in that they have an abundance of waxy lipids in their cell walls, and these extra waxy lipids in the cell walls provides them extra waterproofing protection. So this means that if we are trying to use water-based chemicals or water-soluble chemicals, they're not going to be able to penetrate most of the times through that waxy lipid covering. And then if we're talking about protozoan cysts, these have a special cell wall that prevents entry of many chemicals and shields also against radiation and heat. So again, a lot of different levels of resistance. When we're talking about microbes, we have to consider which microbes we're actually having a difficulty with, and that will help to factor into which types of treatments, which types of chemical agents or physical agents that we do want to actually employ in the overall treatment. If we talk about just some terminology, we have germicides being classified as high, intermediate, or low, depending on their killing or inactivating proficiency, so how effective they are. And when we use that term germicide, notice that it does have the suffix "cide" on it, so that goes along with this is something that is going to kill or permanently inactivate. If something has a high capability, this is going to be able to kill everything. And when we say everything, it is killing things that were at the top of our list, which certainly does include bacterial endospores. When we're talking about intermediate things, this is going to kill most things except for those really resistant endospores. And then if we're talking about low, low is going to kill vegetative, typically non-harmful bacteria, fungi, protozoa, some viruses as well. And this is typically only going to be used for disinfecting items that are going to contact the skin. If we're talking about penetrating through the skin and going to a lower layer, then we probably need to use something that's going to be much more proficient at its killing. So we would use something that's in the high or intermediate category. If we talk about the effects of environmental conditions and how these do tie in with our other microbial treatment methods, temperature is really important. And you can see in this graph right here exactly what effect temperature has. In general, as the temperature goes up, we're going to see more effective killing. And so if you look at the line for the 45 degrees Celsius treatment, we see a much sharper decline in the number of microbes. And if you compare that to the 20 degrees Celsius, which is certainly much cooler, the microbes are not going to die off as quickly. So overall, increased temperature and then also decreased pH, which will be more acidic, those are going to typically increase effectiveness of whatever other treatments we're using at the same time. So we can still treat with chemicals, but then we also increase the temperature at the same time, maybe decrease the acidity, or decrease the pH at the same time.